Hi everyone, in this video we're gonna learn about glycolysis pathway. So what is glycolysis? Glycolysis means the splitting of sugar. So basically it is the metabolic pathway that converts glucose into pyruvate or pyruvic acid and produce energy in the form of ATP. So where does this glycolysis occur? It occurs in cytoplasm, and the source of glucose by glycolysis is mainly obtained from food in our diet. So what is the saline features shown by glycolysis? Well, firstly, it is the catabolic reaction, called as unbanded Meyerhof pathway, catalyzed by specific enzyme and consists of two phases. First one is preparatory phase and second one is payoff phase. In step 1, the enzyme used is called hexokinase. ATP consists of 3 phosphate groups. In this step, one of the 3 phosphate groups from ATP is transferred to the glucose by enzyme hexokinase, thus forming glucose 6-phosphate. The ATP then leaves as ADP. Step 2. Enzyme phosphohexoisomerase. The glucose 6-phosphate is rearranged and is converted into its isomer, which is fructose 6-phosphate, by the enzyme phosphohexoisomerase. Note that the isomerization process is to stabilize the glucose structure.
Step 3. Phosphofructokinase reaction. In this reaction, another ATP transfers a phosphate group to fructose 6-phosphate, which then phosphorylated into fructose 1,6-biphosphate that is catalyzed by the enzyme PFK or phosphofructokinase. Next step, Aldolase reaction. The fourth step is catalyzed by enzyme fructose biphosphate aldolase, which cleaves fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde biphosphate. These two products are isomers.
Okay, so we move on to the step 5, which is Triose phosphate isomerase reaction. The dihydroxyacetone phosphate or DHAP converts to its isomer, which is glyceraldehyde 3-phosphate or G3P, for further metabolism in glycolysis. Thus, the result is 2 molecules of G3P, 3 carbon. So the second phases of the glycolysis pathway is energy payoff, whereas the ATP produced. So the next step is glyceraldehyde 3-phosphate dehydrogenase reaction. Each G3P is oxidized by transfer of electron and hydrogen to NAD+. The NAD+ reduced to NADH plus H+. The product is phosphoglycerate, then react with inorganic phosphate (Pi) from cytosol to form 1,3-bisphosphoglycerate.
Step number 7. Phosphoglycerate kinase reaction. 1,3-biphosphoglycerates are converted to 3-phosphoglycerate. One phosphate group from 1,3-biphosphoglycerate is transferred to ADP, forming ATP through substrate-level phosphorylation. Step number 8. Phosphoglycerate mutase reaction. Each of 3 phosphoglycerate is rearranged by relocate their phosphate group from C3 to C2, forming 2-phosphoglycerate.
Step 9. Enolase reaction. 2-phosphoglycerate is converted to phosphoenolpyruvate (PEP). Water molecule is removed from each 2-phosphoglycerate, which result in formation of double bond. Step 10. Pyruvate kinase reaction. Phosphoenolpyruvate is converted to pyruvate. Each phosphoenolpyruvate (PEP) molecule transfers its phosphate group to ADP, forming ATP. The production of ATP through substrate-level phosphorylation. 1 molecule of glucose can produce 2 pyruvate, 2 net ATP, 2 NADH plus H+, and 2 H2O. That's all for glycolysis.